So it's my great honor and pleasure to have Professor Tom Daniel here. So Tom is actually definitely not a stranger to many of us. He is one of the distinguished senior visiting fellow hosted by IAS, and also he is one of the advisors of biomedical engineering division. Now is part of the Department of Chemical and Biological Engineering. So he's a genius, right? He's among many of the awards he received one of these Master Genius Award, right? For those of you from the North America knows the significance of Master fellowships, and among the many things he received, I just want to highlight two things for those of you who want to publish in like Science or Nature or top journals, and Tom is actually one of the reviewing editors for Science, right? So in case some of your journal, some of the papers that you send to Science that without review and get rejected, you know he's the bad guy behind the thing. And he is also actually one of the scientific advisors of Poissons Brain Science Institute. Yeah, and you know those of you who play watch NFL and baseball game, Poissons, right? Almost several sports teams. In addition to that, he spent a lot of his money in building out the foundation of science and technologies. Okay? One of the actually very visible ones is the Brain Science Institute, and Tom has been serving as an advisory role for many years. So we are very pleased that today he's going to not so much talking about the neuroscience and neuroengineering that he talked about in the past 2-3 lectures. Today he's going to bring us to look at the muscles, the Swell ideas, where I have no idea what he meant, but I'm pretty sure he will tell us what the Swell idea means. Without further ado, please join me to welcome Professor Tom Daniel.
It's always a pleasure to be here and to be teased by me. So I will have my turn with him shortly, as I'm going to do. He doesn't know this, but I'm going to do a demonstration. I want to again thank Komen for welcoming me here. Thank IAS and the department now, the new Department of Chemical and Biological Engineering. I decided to tune this talk more for the title of your new department than neuroengineering. So it's a little bit of chemical engineering, a little bit of biological engineering mixed into a single talk. It's building on a talk I gave 2 or 3 years ago at the IAS, and it's all new stuff from my lab as of a few months ago. So I'll give you a little more background, but before I do, I want to thank my funding. I always start with who's responsible. So the Guggenheim provided partial funds for me to come the first time there. National Science Foundation, National Institutes of Health, the Army Research Office, and Amazon. And Amazon has been funding through a grant a lot of the computing. We use AWS, Amazon Web Services, for a lot of the computing we do. And they do provide grants, and they also do that for graduate students and postdocs separately. So it's a good thing to know about it. You're going to do large scale computing. I have 3 key collaborators for today's talk. The first is Tom Irving, who runs a facility, a national facility at the synchrotron, which is a large high-energy X-ray source. We use his access point to high-energy X-rays, as you'll see. That becomes really critical. He's at Illinois Institute of Technology. My other conspirator is Pekka Hosoi in mechanical engineering at MIT, and she's been working with us on nanofluidics, which I'll take a scale down from microfluidics. And then Simon Spomberg, my past postdoc, who's now in physics at Georgia Tech, and he has been working with me on molecular mechanics and also all of the work I'm going to talk about today. So these 3 are my key collaborators, and then the really most critical part are the members, my happy, smiling lab members that are my postdocs, Itai, Julie, Tanvi, Colin, and Dave, and I can go on down the list. wonderful graduate students and undergraduates working in the lab. I just want to call out, I put halos over the ones that were mostly involved in today's work. And that is Julie Cass, who did a lot of the simulations. Dave Williams is the postdoc who began it. He is now a researcher at the Allen Institute of Cell Science. So those of you who are more interested in cellular biophysics and cellular dynamics, they're actually created a new institute. They're hiring research scientists all the time. Joe Powers and Sage Melligan have been working on computational aspects of today's talk, and Brandon Pratt has done some earlier work on muscle. Okay, let's start with various introductions. Muscle is a really interesting material. We call it active matter, active regulated material. Active matter has received the attention of physicists, and in fact, it was because of interest in active matter as a novel kind of material that drove the Army Research Office to challenge investigators to look for really interesting materials which don't follow the normal paradigm of passive materials that we think of in things like traditional condensed matter physics, right? People have referred to something as interesting as a flock of starlings as essentially behaving like active matter. That is, these individuals have local rules. They're using energy. That use of energy and local rules gives rise to these emergent dynamics which move in really, really interesting patterns and behave as if they are a material, right? Active matter generally consists of the following rough ingredients. It's matter that's composed of a large number of agents. And in the case of muscle, those agents are molecular machines, okay? The agents consume energy. They interact with each other. They move. These are also true of starlings and molecules, right? And they generate forces, as do starlings and molecules, right? They're intrinsically not at equilibrium. They're consuming energy at all times. So this is not an equilibrium dynamics problem. This is a non-equilibrium, energy-consuming, multi-agent, emergent property problem. And this is sort of interesting. What are the underlying physical principles that govern the behavior of something as complex as muscle, right? And collectively, in muscle, these agents produce forces that may act as an actuator. So I can move my arm, control my motion. They can act as a spring. Muscle can act in a way that is neither actuation nor resisting actuation, but it's more elastic in its dynamics. And muscle can also behave in a way that's like a damper. And it can do this by merely changing the timing in which we activate muscle. So to be clear, as I'll go over shortly, muscle is made up of many, many millions and millions and millions of interacting particles. These particles consume energy. That consumption of energy involves interactions between particles, and those interactions lead to forces. And those forces lead to motions, okay? And those motions lead to a great image from TC's poster. So our goal in this part of my lab group is to understand the biophysical principles underlying the physics of muscle. So treating muscle as a physics problem, as a reverse engineering problem, okay? And we're particularly interested in two things. Let me go back a slide, if I can. First is, how is force regulated and produced in muscle? You would say, well, surely we have nailed this problem. We completely understand this. Muscle has been a subject of intense investigation since the 1950s. There remain a significant number of open problems, as I hope to indicate shortly. And the second is, how is energy transport managed in muscle? It's a highly organized lattice of interacting dynamics, of interacting proteins. Okay? So just to give you a frame of reference from an engineering standpoint, I just want to compare a few things here. And I have on the left an engine, a battery, and biology, okay? These are my points of comparison. All of them consume some form of energy. All of them undergo or produce some actuation, which leads to some motion and forces, and those motions will propel something. So I can have a gas-powered engine whose thermodynamic efficiency in an ideal case is about 56%. Gas-powered engines have existed in robots. They do very, very poorly, okay? That said, the energy density of petroleum products is quite high. Okay? In contrast, a battery-driven machine tends to run at generally very good efficiencies, right? But the energy density is quite low. So for example, if you were a person at HKUST in the very famous quad rotor program here that DJI grew out of, batteries are heavy and they're a serious challenge to development of micro-air vehicles, as we have talked about in previous talks. So in gas-powered engines, the thermodynamic efficiency is sort of poor, but the energy density is very, very good, right? In battery-powered systems, the energy density is very, very good, right? Or rather, the efficiency is good, but the energy density is quite poor. Battery technology is still lagging behind much of what we worry about today. And in biology, actually, the efficiency is pretty good. It's not as good, right? But in fact, it's good, and the energy density is very good. So carbon-based energy density is, in fact, an excellent source. So biological systems tend to operate really well. And in muscle, there's a really interesting phenomenon that it just scales so easily. So for me to make an electric motor running a robot, it is a hard problem to simply make a bigger robot with that little motor, okay? But muscle, in something as small as a planktonic organism, that's at best a millimeter long, you would be hard-pressed to distinguish the cellular differences between their muscles and those in a whale. You simply scaled it up by making them much bigger. It's the same machine. It's the same organization. If you look at the cell, you can't tell them apart. A skeletal muscle cell in a plankton looks just like that in a whale. It's just that there's that much more of it. We have a very, very hard time scaling motors, okay? So these are just some observations. I want to now take a dive down and tell you what I want to do in today's talk. So my motivation is to do the sort of the reverse engineering of muscle and to ask, what are some of the design rules? What are some of the physical principles that are going on as we convert chemical energy into mechanical work, okay? So I'm going to give you a very little bit of background on muscle, just how it's built. So not everybody in here knows the organization of muscle, so I will briefly review that, okay? I then want to go over very briefly some past computational analysis we had done, how we approached this using AWS and whatever services we had. And then I want to dive down into a really interesting challenge is how do I make both predictions and measurements at the scale of nanometers and angstroms in live, active muscle? How can I actually make measurements at such a tiny scale, at the protein scale, while the muscle's alive? What tricks can we use to do that? And this is some new work that we are writing up just now. Then I'm going to take a dive into two new areas that have been completely ignored in the biology and physics of muscle. And one area is the fluid forces on molecular motors. So I'm going to tell you right now that molecules do not behave in a vacuum, but our models treated them as if they did, okay? That's an interesting issue. So proteins operate in a fluid medium, but the models that we have built in the literature have ignored that fact, okay? There's some good reasons why they ignored it, but it bothered me as an engineer. As somebody who was trained in fluid dynamics, it really bothered me, okay? And then I'm going to do one more thing is I'm going to talk about diffusion and convection, fluid dynamics coupled with diffusion and how those might affect the dynamics at the nanometer scale, okay? So that's what the outline of my talk is. I'll come back to this periodically. But I'm going to conclude, I'm going to tell you what the punchline of my talk is, that muscles move in such a way as to not only generate motion and force, but also to fuel themselves with energy as they're doing so, okay? That's my take home. So I go back to the 1950s. This is my hero. This is whose work I have read every paper he's written. This is Andrew Huxley. He died only a few years ago. Andrew Huxley was known for two very, very famous things. One is the Hodgkin-Huxley equations, that's Huxley, for the fundamental principles of action potentials, how neurons can propagate signals. That's number one, right? And number two was something called the sliding filament hypothesis. That is how muscles and proteins interact in muscles, converting chemical energy into mechanical work. The obituary for Huxley in the New York Times read as Andrew Huxley, a British scientist from an illustrious family whose boyhood mechanical skills led to a career in physiology. Okay, that is what they wrote. And in quotes, he called it the mechanical engineering of living things. That was his view of the world. And it's one that I've certainly enjoyed. And he won a Nobel Prize for explaining the electrical and mechanical basis of bodily movement. And what is so important to know about what Huxley did is he said, in order to explain how signals are propagated along neurons, he had to posit the existence of a particular behavior of a protein. That behavior is called a voltage-gated porosity or channel of a protein. Nobody even knew of such a thing, but he had to posit the existence of something that was later found out to be true, right? It took incredible insight. And it was that same insight that he had to posit the concept that proteins cleave energy from ATP and undergo conformational changes in generating force in muscle. It was amazing insight into having to posit things that were later found to be true. He felt, by the way, that he should have gotten 2 Nobel Prizes. And I actually agree with him. I think he should have gotten the one that he did get for explaining nerve conduction, action potential, but I also think the sliding filament hypothesis and the basis by which we generate motion was warranted a prize as well, particularly given the insights he had. So I'm going to focus today on the latter, on the sliding filament hypothesis. Even though a lot of my lab and certainly visitors from HKUST in my lab worry about this other part, right? I want to focus on this one, and I'm highlighting the one word. You can barely see I've highlighted the word sliding, okay? So I'm going to go over this, and we're going to challenge Huxley a little bit. Not a lot, just a little, and see where this takes us, okay? So let's get to the meat of the issue, as they say in muscle biology. Muscles, as you know, are arranged longitudinally. So a muscle, my biceps, runs from an origin here to an insertion on my arm, on my ulna here. And that muscle consists of a bunch of cells, okay? And each cell, right, here's one of these cells right here, blown up. So this whole thing is a cell. That cell has a membrane, okay? And the membrane is not perforated, but it's indented with conductal surfaces, okay? Critically, it's a cell with a membrane, and a cell with a membrane has, under whatever shape change you do, it's constant volume. There's no water going into or out of the cell that we know of, okay? And to demonstrate that point, I promised I would use Professor Singh here, okay? Could you please stand up? This is for giving such a wonderful introduction. Please face the audience, okay? Professor Singh has muscles. It may not be obvious, but and, sorry, when he contracts, go ahead, contract your bicep. His bicep shortens and it gets wider. Not by much, okay? Yeah, I feel better now. So, as the muscle cell shortens, of course it gets wider because the cells have constant volume. This whole lecture was all about this. I can go home now. But that turns out to be more profound than you think. Okay, and I'm going to explain why shortly, because this becomes super critical that we begin to think about, wait a minute, if the muscle shortens and it gets fatter, what does that actually mean inside the cell? What does that actually mean to the dynamics of the interacting proteins? Okay, this is, as I told you, what we're talking about is the sliding filament hypothesis, not the expanding filament hypothesis. Okay, so let me go on a little further. So the cell has constant volume. And inside the cell are mitochondria that produce energy. Largely, there's creatine phosphate, phosphocreatine kinase, and other enzymes of critical importance within the cell. Okay, and within the cell, you have these subcellular elements. They're called myofibrils, and they are not cells, but originally they are formed like a syncytium of cells, right? But they're highly organized units that themselves, here's a myofibril right there. So this is one cell, it consists of many myofibrils. The myofibrils are solid, or not solid, but densely packed arrangements of proteins involved in force generation, okay? And they're surrounded by a variety of regulatory structures as well as energy sources. But inside them are only contractile proteins. There's no mitochondria inside the myofibril, okay? And to know a little more about this, the myofibril is made of two interacting protein types plus a third one that we're very, very interested in. In blue, we have something called the thick filaments, and they're called that because they're appear thick. And you'll see why in a minute, and nicely labeled. This is some of the nicest biological labeling I've ever had. The thinner ones are called thin filaments. And these are made up of a protein that you'll see shortly called actin, to which motor proteins bind. I'm going to go over the details in a second. Also shown here in green is another protein. It's the biggest protein in biology. It's called titin. And titin actually still serves a somewhat unknown role in dynamics of muscle contraction. The largest protein in the world, and we still don't completely understand how that works. There are lots of interesting titin mutations. There's also critical proteins on the edges of this that I'm extremely interested in called dystrophin, which is a protein coded by the dystrophin gene family. And that's involved in controlling radial mechanical force transmission in muscles. Also extremely poorly understood, disorders of the dystrophin gene lead to muscular dystrophy, which is a profound movement disorder. It's a loss of effective contractility. So if you look a little deeper now, here is that thick filament with these motor proteins. These are myosin molecules extending outward. There's the thin filament made of these actin monomers made into a beautiful double helix. And they are anchored, here's titin anchoring off to what's called the Z disc, which is made mostly of a protein called alpha-actinin. I don't actually care that you remembered the names. I'm going to go over them again in a bit, but there's sort of a critical dynamic that goes on here. So let me see if I can explain a bit about the dynamics. Here I have the thick filament with the motor protein extending off, binding onto a thin filament. And I'm going to do a little bit of pantomime. But what happens is that the thick filament with that motor protein extending outward, that motor protein is actually an enzyme. And what that enzyme does is it cleaves the terminal phosphate off of ATP, adenosine triphosphate, into ADP and phosphate. And I'm just going to play that movie again, but let me set you up for it. ATP is a molecule of a modest size, a few tens of angstroms, right? When ATP is hydrolyzed into ADP, adenosine diphosphate, and the separate phosphate, that cleavage of that terminal phosphate expands the molecule because that double bond holding that terminal phosphate actually holds it quite closely to the subterminal phosphate. On cleavage, in fact, ADP and phosphate occupy more space than ATP because of the cleaved terminal bond. That space occupancy is, in fact, transmitted into a distortion of the molecule. Okay? Interestingly, adenosine diphosphate, ADP, and ATP aren't that different in size because of that very tight terminal phosphate group, okay? So what happens is, on cleaving that, the molecule is distorted. On binding to the thin filament, that distorted molecule, now it's favorable for the phosphate to fall off. And on that falling off, that distorted spring-like stretch from the hydrolysis is transferred into a mechanical motion, into a distortion of the molecule, okay? Now I'm in this distorted state. I have ADP, which is very close in size to ATP, and in this distorted state, I can favorably exchange this. It releases from that thin filament, hydrolyzes the ATP into ADP and phosphate, and returns that cycle. So the key idea is that the hydrolysis of ATP is, in fact, expanding the molecule, stretching locally internal springs in the molecule. That's the idea, okay? And as this movie shows, you bind with ADP and phosphate. The phosphate falls off, right? That undergoes this distortion in a very, this is a cartoon. ADP comes off and ATP is substituted, and you have a release of that. And you can repeat this cycle. As long as ATP is present, you can repeat that cycle. And importantly, the rate at which this motor molecule can operate will clearly depend on phosphate and ADP and ATP levels, just like any enzyme kinetics will, okay? So far, so good? So what we have is this idea that ATP powers the conformational change. In this weakly, here's ADP and phosphate. It binds, the phosphate pops off, it undergoes a distortion, dragging the molecule past it, and then ATP is substituted. That's all. That's the chemistry. There's chemical engineering that just happened. And that happens in a three-dimensional lattice of thick and thin filaments. So I showed you a picture of them just overlapping in a cross-section. In fact, it's 3D. And in fact, it's much more packed than I'm able to show you here, okay? So far so good. There's still background here. As muscle shortens, okay, these filaments, these thick and thin filaments, the motor molecules drag them past one another. They slide past one another, right? And if you start them very, very far apart, at a very elongated muscle, right? There's very, very few motor molecules extending from the thick filament to the thin, so you get very low forces. As these thick and thin filaments overlap more and more and more, the forces get greater and greater until you have maximum number of overlapping thick and thin filaments. The maximum number of cross bridges are recruited, and then that declines as the thin filaments begin to overlap themselves. This is classic result from Gordon, Huxley, and Julian. Demonstration of the sliding filament hypothesis. That when the filaments aren't overlapping very much, you get very low force. When they're overlapping maximally, you get the most force. And when the thin filaments begin to overlap with each other, you get less force. I mostly agree with this. But as Professor Singh demonstrated, right, in cells with constant volume, their diameter has to change, right? If I shorten a muscle, it's got to get fatter. What happens to the filaments? If the filaments go from long to short, long muscle to short, do they spread apart? And if they spread apart, do motor molecules have to reach a greater distance to bind? And as things get shorter, is it just harder and harder for the motor molecules to reach across the gap to the thin filaments? Wouldn't that be interesting? So what I had here is that motor molecule, sorry. So if this distance increases this way, right, I will decrease the likelihood that that motor molecule could find a binding site, by definition, okay? So I'm going to, again, sort of quickly go over this because this is something we did a while ago, and just enough of it to give you an idea of where we were and what drove what we did next. So Dave Williams and Joe Powers modeled this process as a Monte Carlo system. And this is where we were using sort of large scale computational models. Okay. I so insulted him that I knew he had to leave. Okay. So this is where we used AWS. So we started with a backbone of a thick filament and represented that as a bunch of springs. Okay. And then extending from the thick filament are these motor molecules that were represented as both torsional and linear springs. Okay? That those cross bridges, those myosin motor molecules, faced a thin filament, which is a double helix of binding sites. And those have a periodicity that we know from X-ray diffraction, as I'll show you. Okay? And those also are represented as a set of springs. And then all we did is simply use a Monte Carlo simulation and use appropriate estimates of the kinetics of these molecules to do the following. We can specify how much the filaments are overlapped. That is, how many motor molecules are facing each other. This is just a computational term we specify. We can specify how far apart they are. And each motor molecule can be subject to thermal fluctuations that are set by the temperature. And we know the energy state associated with it. So with a Gaussian energy state, we can actually drive these motor molecules around, and eventually they will be within a certain distance, a Debye length of a binding site, and they will bind at whatever distortion they may be at, driven thermally. Okay? And the thermal driving is just throwing a random number generator over and over again. For every molecule, there's many molecules in the system, right? In 3D. Okay? We can specify the spring constants, and these are actually known in the literature. How stiff the thick filament is, how stiff the thin filament is, what the torsional and elasticity characteristics of the cross bridges are. These are all now well known in the literature, and we're simply using the values. A lot from optical tweezer experiments and the like. And we also can specify probabilities of binding, that is the cross-bridge kinetics, the rate at which cross-bridges can hydrolyze ATP. These are things, again, we're drawing very heavily on the literature. So using a Monte Carlo simulation, we actually have to do inverse optimization because this isn't, the angles of the spring introduce transcendental functions that require, there's no linear solution to this, so it's just an optimization method that we've used here. And from that, we calculate your probability of binding, your distortion when bound. Given a distortion, if we know the spring constants, we know all the local forces. Okay? If we know all the local forces and all the distortions, we can calculate the force generation. And here, for example, are just turning a muscle on with two different levels of activation. Okay? This is just simulation result. Okay. So the thing I want to do now is carry this one little step further and say what we did is we measured these sort of asymptotic, what we call isometric forces. That is, we held the muscle constant just in length at any particular length, right? Any particular amount of overlap, and monitored the force. Okay. And so what I'm plotting here is a bit of a challenge. So let me work you through the axes here. This axis is the force generated by the simulation, right? The thousands of motor molecules. It's in piconewtons, all right? This is the sarcomere length, which is just how much overlap there is. So at very long sarcomere lengths, there's very low overlap. At intermediate sarcomere lengths, there's 100% overlap. And at very short sarcomere length, the thin filaments overlap with each other, okay? And what we find, lo and behold, is we get exactly what Huxley got, right? Over and over and over again, okay? Now let me show you the other axis. This axis is something called the lattice spacing. So I can take muscle at this much overlap, and I can have the filaments this far apart, or this far apart. I can swell the filaments apart, just mathematically, just in the simulation. And let's think this through. So at very large lattice spacings, this curves down and I lose my ability to generate force, and that's because my probability of binding gets very low when the distances in the radial direction are very large, okay? At intermediate, I get optimal force generation, And at very, very tight spacing, I get very low force generation because the molecules are in a position that's not favorable for generating force. So they're more angled upward, so to speak, not generating force in a favorable axial direction, okay? Well, I don't know what muscle does. It could be that this is the only axis that matters biologically, that only sliding filament happens, that there is no expansion of the lattice. That is, when muscle shortens, it just stays its all same length, right? Now, the muscle cell may get fatter, which will present a problem in terms of conservation of mass. Or it could be as the muscle gets shorter, in fact, the filaments expand, right? So there's constant lattice spacing or constant length, right, with varying lattice spacing, or something in between, constant volume, right? We just don't know. So these spatially explicit models, by the way, don't scale very well. We've run up against sort of the computational limits, and so we are looking to collaborate with data science and some data analytics to how to make what are called reduced order models so that we can scale these up. But right now we're stuck. Okay? This is as good as we can do. But what happens biologically? What do the proteins do? So this requires that we make a measurement of proteins in contracting muscle and their spacing. So this is where, so I'm going to start out with a hypothesis. So now we're going to get into the serious meat of the talk here. That is, the lattice spacing of filaments would expand inversely with the square root of the length of the muscle. I only said that because that would be the hypothesis of constant volume. So if I have a cylinder of contractile proteins and they shorten, if the area expands in direct proportion to the shortening, then in fact volume is conserved. And if it's an area that's expanding, then the spacing has to go as the square root of the length, okay? That was all. This is the hypothesis, right? And what we do is we use a model organism. I can tell you a little more about this. This is the hawk moth Manduca sexa. Its genome is now known. We can, it's got a fabulous neural system to study, as certain members of this audience could tell you. But it also has, as I'll point out, a muscle that can be electrically controlled very precisely. That's important for us. And it turns out to behave like cardiac muscle, which is another reason why we chose this, okay? We take this to something called the synchrotron in Chicago, Illinois, which is a high-energy X-ray source. And we operate on one of the sectors of this, which has it's called low-angle or small-angle X-ray scattering, okay? So we use Manduca because the muscles can be activated what we call synchronously. I can time it, I can send an electrical pulse to the muscle, and it will generate an all or none contraction. Okay? That's number one. Unlike vertebrate muscle, unlike your skeletal muscle, those require a volley of excitation signals. These are one-for-one correspondence between a motor neuron excitation and the actual contraction. That means I can time their contractions extremely precisely. Okay? Second, they have cardiac-like behavior. They're ideal for X-ray. It turns out that Drosophila, wonderful as it is, tends to get toasted in an X-ray beam of this intensity because they're so tiny, right? These animals have about a cubic centimeter of muscle that is about the right volume, so we don't have too much heat generation from the X-ray, okay? And the organization of the proteins are extremely, extremely organized. They're almost paracrystalline. Many of you have heard of X-ray crystallography in protein. Protein X-ray crystallography was a way we get three-dimensional geometry of proteins. But you have to crystallize the proteins to get that. In muscle, the proteins are semicrystalline or paracrystalline. So in fact, you can get under this low angle X-ray scattering, right? You can actually get really, really good pattern from live protein, okay? So what we do is we take the muscle, we can control its length, we can control its activation, right? We take the head, wings, we take most of the animal off, and we place the muscle in a device where we can measure forces, we can control the length, and we can control the activation, okay? That device is then centered into a beam at the X-ray facility, right? And that beam then generates something called a diffraction pattern whose data we can mine, okay? And the intensities are, in a sense, the Fourier transform of the spatial distribution of the proteins. So what you're getting is not an actual movie of the protein, you're getting a movie of the Fourier transform of the protein, okay? You're getting a movie of the protein shape distribution, okay? In a sense, all right? So just to get you oriented, here is the thick and thin filament array that I showed you earlier. We call this a sarcomere, right? From Z disk to Z disk. There are thick filaments in blue, thin in red, okay? In this plane, right, we are seeing pattern that's reflecting whatever periodicity that exists in this axis here. And in this axis here, we see the manifestations of periodicity or regular structure in this axis, okay? So we get two axes of information. I'll give you just a little more orientation. Here is what we call the 2.73 and 2.9 nanometer reflections, okay? It doesn't mean much, except these are the twists of the two sides of the thin filament. That's the periodicity there, okay? So in fact, we're picking up thin filament twist, thick filament twist. These are the helical arrangements of these proteins in this axis. Down here, we're getting information about this sort of spacing and periodicity, okay? It's really hard to see, so I'm going to do a little blow up of that. Here again are the now thick filaments in red. I apologize for the color change. Thin filaments surrounding them hexagonally. The spacing between the same lines of thick filaments is called the D10 spacing. And it is related to the space you measure in the Fourier transform, right? And inversely so, or on the X-ray image. And the wavelength of that particular X-ray beam, and the distance to the sensor, the optical sensor we have, okay? So the farther apart those spots are, the closer are the spacings of the filaments. Okay. So we're oscillating the muscle lengthwise, and to be oriented correctly, I'm doing this now on the muscle, okay? Along its length. And as its length changes, we can monitor the force, we can monitor the length, but we can also monitor the spacing in real time. And we're doing this at very high resolutions. We're oscillating these muscles at 25 hertz, and we're going to get sub 25 hertz, sub 40 millisecond resolution on an X-ray diffraction, okay? And so a blow up of that here, that is actually now reconstructed. I mean, we had to go mine each image separately, right? But this is a movie showing that it turns out the muscle's angling a bit, but it's also expanding and contracting. So the sliding filament hypothesis is mostly correct, but not 100% correct. That is, filaments do slide. There's no question about that, right? But they're also expanding and contracting. The question is, are they doing it according to constant volume or something else? So I want to remind you of something called Poissons Ratio, okay? So with length changes in radial spacings, Poissons Ratio measures, in a sense, that relationship between when you pull on something, does it get thinner? And if so, by how much, right? So a constant volume elastic solid, right, has a Poissons Ratio about 0.5. That is, the radial dimension changes as the square root of the length, which is what our hypothesis was. So our hypothesis that I said is muscle behaves as a constant volume material. That is, its Poissons Ratio is 0.5. By the way, I just thought this was great. It's a quote from Semyon de Poisson. He says, "Life is good for only two things, doing mathematics and teaching it." I liked him. It's possible, however, to pull on a structure and have it get fatter, to pull it outward and have it expand radially. And this is called an auxetic structure, okay? And here's an example. If I pulled on this triangular framework in the axial direction, right? As these rotate outward, they expand in the radial direction. So that's a structure, in fact, which when you pull it one way, it expands, right? Or if you push it, it would contract. So it's called an auxetic structure. It has a Poissons ratio that's negative, right? I mean, you can build these, and they have been built, and they do pretty much what you thought. They're frames, though. So it's really hard to think about a material as a frame, right? This is a frame that has, whose linkages do pretty much what you would expect when you pull on it. It's auxetic. But is the myofibril, that subcellular bundle of thick and thin filaments, is that a frame or a solid? Now, how is it behaving? So let's keep going. Here would be the length that I've given the muscle, right? And I have several ideas. One is I have the sliding filament hypothesis, right? That is the lattice spacing, the spacing of the proteins in the radial direction is constant, right? In time. And if you plotted lattice spacing versus length of the muscle, it would just go back and forth like this. The lattice spacing is not changing. That is sort of the background of the sliding filament hypothesis. The other option is the constant volume one. That is, as the length changes, the lattice spacing changes inverse to the square root of that length, right? In which case it would have this modest slide up and down on the lattice spacing versus length graph, oscillating back and forth along that line. Those are two possibilities. We don't know. So let me show you our data. Okay? Here is the length and time. That dot represents when we stimulated the muscle, right there. We can vary when we do this in the cycle, and we deliberately picked a model system in which we could do that variation. All right? Here is the force, and time is coming up, and then it pops right after we do the stimulus. And here is a plot of the force versus length for that muscle. And you know what? It looks like it's not going to be very interesting, but that's force versus length. That's not lattice spacing versus length. This is something called the work loop, and this muscle is behaving fairly elastically, okay, at that particular phase of activation. Here again is the constant lattice spacing versus time. Here's the constant volume versus time, and here is our data at 1, 2, 3, 4, 5, 6 time points. The gray is our standard error bars, okay, for many, many, many cycles, okay? And it's neither the green nor the red. It's something other than constant lattice spacing or constant volume. In some way, when the cross bridges are recruiting, they are either restricting or governing the radial expansion and contractions of the lattice. And to some extent, you would expect that in 20/20 hindsight. That is, as the cross bridges bind, if you wanted the lattice to expand, you'd have to put energy into expanding or distorting the cross bridges or contract, right? So to some extent, I can look back and go, but of course, but I didn't know this going in. And here's the interesting plot. Now what I've plotted here is lattice spacing versus length. This is not force, this is the lattice spacing versus length. So I'm plotting the blue against this plot here, right? Here is what I expect for constant lattice spacing. As I go back and forth on muscle length, nothing. Here's what I expect for constant volume. As I go back and forth, that. And here's what I get for the actual muscle. And it's sometimes as the muscle shortens, the lattice spacing expands. I like that. And sometimes as the muscle shortening, the lattice spacing is contracting. As the muscle shortens, it's contracting inward radially, dependent on when I activate it. So this is a very odd material. This is a time-dependent Poissons ratio if you're an engineer, which is something we don't build, we don't think about, right? We're about done, and I'm so far from done. Here is the lattice spacing loop for 2 different phases of activation, showing, in fact, just by changing the timing, I change the apparent physical I'm going to quickly summarize where we are now, because the data are new and this is what we're writing up, but let me tell you where we are now. If the lattice spacing is not constant, right, or it's not constant volume, there have to be fluid exchanges going into and out of that lattice of protein. And if there are fluid exchanges going into and out of that lattice of proteins, there are two things going on. There's fluid shearing, I cannot help it, right? And there's potential substrate flux. So as the sarcomeres are going like this, I can be pushing fluid out, pulling fluid in, pushing fluid out, pulling fluid in. Can in fact be a mechanism by which I told you there's no ATP available on the inside of the lattice. It's all has to come from exterior to the lattice from the mitochondria. So our idea is that this dynamic is a way of providing both flux of energy and mitigating the fluid shears. So the space is very, 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 very packed. And there's nanometer scale gaps in there. And this is a level of fluid flow problems that we have rarely had to deal with. But I can't help it. The kinematics, just how the proteins move, mandate that there has to be fluid motion going in and out of this. And it's completely ignored in the literature. There are approximately 0 papers on it. Okay. What are the flows? What are the viscous forces? So as those filaments contract, if it's just sliding, I can guarantee you have to force fluid out. I can guarantee it, right? In Drosophila, what real-time X-ray diffraction there was suggests there's no appreciable radial motion, but it turns out their length changes are so tiny that you wouldn't be able to measure the radial motions if they were there. Whereas in our system, Manduca has these significant radial motions. So to address this, I'll just go really quickly. Sage Malingen and Pekka Hosoi, our collaborator at MIT, and we built up a mathematical simulation of cross bridges in thin filaments and thick filaments consisting of gazillions of little tiny spheres known as Stokeslets. Has anybody heard of a Stokeslet in here? Okay, that's good. How about Stokes? Great, what is Stokes? It's Stokes flow, exactly, yeah. So no inertia, purely viscously driven flow, which is what we expected at this spatial scale. Excellent. So there's this numerical method called Stokeslets. You point singularities of Stokes solutions. And you can solve the flow equations for very, very complex geometries by representing things as points of tiny, tiny momentum sources that obey these very, very low, tiny scale flow problems. Here's a flow field around a spherical cross bridge that we predict. And it has drag on it. It's not very big. In fact, the drag is a tiny fraction of the force that cross bridge can generate. But I'm going to skip a bunch of slides in the interest of time and tell you what we've done. We've solved for the entire flow field in the lattice as it's sliding, okay? And that flow field allowed us to calculate how much energy is associated just with viscous shearing. And in a sarcomere, it's about 200 ATPs per second. Doesn't mean a lot, except each cross-bridge cycle consumes 1 ATP. So just to overcome flow requires energy. But that should not be also not a surprise, right? That flow is not free. Our problem, and I don't have an answer for this, I'll leave this to the brilliant bioengineers in the audience, is we have no idea what the properties of the fluid are in the spaces around the filaments. We can say that they're at least as viscous as water, but probably more so. But I don't have any data on that. So we're guessing at this point what that has to be. We can bound it, but we don't really know. Okay. But for a set of cell types, if it's the viscosity of water, in some cells, the cytoplasm is 3,000 times more viscous than water. and some, a mere 100 times more viscous than water. So cytoplasm is more viscous than water, largely because of the large proteins in it, right? And it's fairly thick with solutes and protein and the like. That makes it more viscous than water. But we just don't know what's in the lattice. And that's our biggest thorn. I just need to do this one more thing because it's what my newest postdoc just finished, and it's so good, I apologize. How does stuff get into there, okay? How, when this muscle is contracting, how does energy get into the core of the thin and thick filaments? There is no source of ATP on the inside. It has to get in. And people have done some estimates of this by looking at diffusion, right? But we now have something called the diffusion equation. The change in concentration in time is proportional to this spatial gradients of concentration, and the flux due to whatever flow field we have. And the question is, what's going on inside a muscle, inside the lattice? And so Julie Cass in my group numerically solved the diffusion equation with flow, right, for a myofibril, right, with a certain radius and length, specified boundary conditions for concentration of ATP on the outside, and starts it initially empty and lets it go. And lo and behold, these are the diffusion coefficients estimated from the literature. It takes quite some time actually for ATP to get in because it's big relative to the gap spaces available in there. It takes, this is like 1/10 of a second here, there's no ATP on the interior, right? It took something like a full second to get reasonable levels of ATP to the interior of just a myofibril. I should stop, but she went on to solve the whole equation with flow and found that when you oscillate a muscle like this and generate that flow field, she solved the flow field, which I've shown in streamlines here and in a movie here, okay? There's the flow field for this chunk of the muscle here, right? And found that on the interior with no flow, with just diffusion, right, you get a relatively low concentration, but just adding oscillatory flow and taking the average concentration, right, you do better. You always get more substrate in. And what we found so interesting about this is the flow is completely reversible. If I push in this way and I pull out, every stream will go in and go out exactly the same. So initially you thought, well, why would purely oscillatory motion generate any net advantage if the flow is completely reversible? And the answer is the flow is reversible, but the equation itself, the full equation, is nonlinear because of the diffusion term plus the reversible term, right? And that nonlinearity leads to a favorable concentration due to oscillatory motion. We can tune the dynamics, we can tune the diffusions to do better or worse, right? So I think now I can end with the following, that these motor molecules, this whole process generates some flow. That flow can help deliver ATP, right? That ATP can power the motion, which you need to drive the cross bridge, which generates force, which generates filament motion, which generates sliding, which generates flow, and that flow costs energy, but it also brings energy in. And that's sort of an interesting juxtaposition that I am unaware of anywhere in the literature. So there's lots of room for collaboration on this. There's lots of unknowns. We don't know if the net cost of the shearing is offset by the advantage of the flow. We don't know that yet. We don't even know what patterns, nanoscale patterns exist because with embarrassment, I've left something out. ATP is consumed, there's a reaction rate. And the only reason I bring this up is the full equation, which we're working on what this reaction rate looks like, is a diffusion reaction equation, which has been heavily studied decades ago by Alan Turing in demonstration of how you can get pattern spatial temporal pattern dynamics by coupling diffusion and reaction. We know we have this in the system, but we haven't been able to really nail what the reaction rate is. So there's a lot we don't know. There's a lot we're working on. And I went way too long. So I'm going to say thank you to all of you. And I'm around for days. So if there's somebody who likes nanofluidics, reaction diffusion equations, macromolecules interacting with fluids, I'm more than happy to chat separately. Yeah?
What's the case with smooth muscle tissue that's not striated? Does any of this apply?
Wow, that's such a good question. So smooth muscle is, so I thought we really understood skeletal muscle. We really don't understand smooth muscle very well. So smooth muscle still has the same fundamental phenomena. Filaments do slide. One of the key differences are there's no regular Z discs, right? So the regularity of structure is nowhere near as great. But in fact, they're still sliding, and in fact, in smooth muscle cells, they still have to radially expand, right? So the general idea applies. There are some different myosin enzymes and different protein dynamics, but the general ideas are the same. That's a great question, Alex.
Lecturing, and that's all. Thank Tom Daniel.
Thank you all.